A very good morning, everyone. I'm Juan Zhou from Duke US Medical School. So in the next 15 minutes, I'd like to share with you our recent work on functional connectivity in aging and neurodegenerative disorders. Here are the T1-weighted structural MRI of patients with 5 different neurodegenerative disorders. As you can see, each have very distinct atrophy patterns. From the top, Alzheimer's disease, in short AD, usually lose their episodic memory and visual-spatial function, cause atrophy in this posterior hippocampal-temporal, posterior cingulate cortex, and bilateral parietal network. In contrast, behavioral variant frontotemporal dementia, in short BVFTD, has atrophy in this anterior cingulate cortex, frontoinsular network, and they usually come with social-emotional dysfunctions. Based on the evidence from neuroimaging, neuropathology, and animal models, neurodegenerative diseases have been thought to relate to network-based neurodegeneration. In 2008, when I was a postdoc in Bill Seeley's lab, Bill led the team to systematically test this network degeneration hypothesis in living humans using network-sensitive neuroimaging method, which is resting state fMRI. The specific question we ask is that whether the syndrome-specific atrophy pattern you see in disease mirror the brain functional connectivity networks or intrinsic connectivity networks, ICN, in healthy controls. The regions in blue are the atrophy pattern in each of the 5 disorders. We picked the most atrophied region, for example, right frontal insula for BVFTD, and performed the seed-based functional connectivity mapping. The resulting ICNs are highlighted in yellow. As you can see, there's a great similarity between the 2 maps, atrophy and ICNs. This confirms that each disorder targets a very specific network in the brain. The regions wired together within the network, they file and shrink together. And this work opens up interesting questions on selective network vulnerability along the disease progression. Jack, Ewers, and colleagues proposed and refined the major AD biomarker cascade models from preclinical to dementia stage of Alzheimer's disease. Resting state fMRI is one of them. Today, I'm going to focus on 2 specific questions using resting state fMRI. First, can resting state functional connectivity detect symptom-specific changes in dementia subtypes? And second, can we track these functional connectivity changes longitudinally in pre-dementia stage? Let me begin with the first evidence in the dementia stage. As you know, AD targets the deformal network specifically. This is supported by increased amyloid burden, atrophy, and hypometabolism in the deformal network. In 2004, the pioneer work by Mike Grisius and colleagues found that there is a reduced functional connectivity in deformal network in Alzheimer's disease patients. One step further, we have shown that mild AD and BVFTD patients actually feature divergent functional connectivity in the deformal network and salience network. Compared to the healthy control, BVFTD patients have reduction in the salience network, while AD patients have reduction in the deformal network. But importantly, when you look at the mild AD, they actually have increased functional connectivity in the salience network, including pregenual ACC and left ventral striatum. And BVFTD patients have increased functional connectivity in the angular gyrus of the deformal network. Such divergent functional connectivity pattern correspond well with the symptom deficit profile of these 2 disorders. Given the multidimensional nature of the neurological symptoms and the complexity of brain networks, it is important to characterize multiple large-scale brain networks and the inter-network functional connectivity. The human brain, as you know, is highly modular and distinct. There is high functional connectivity within each network and low connectivity between the network. This is to support the specialized and integrated information processing. The task-positive network, like executive control and salience network, and the task-negative default mode network are often anti-correlated or segregated. The salience network is closely connected with subcortical structures as well as executive control network. We hypothesize that such network coordination is differentially disrupted in different dementia subtypes and may lead to cognitive or social emotional symptoms. To test this hypothesis, our team decided to systematically test the whole brain functional connectivity in AD and BV-FTD based on a Singaporean cohort. This is the fresh data from the lab after accumulating BV-FTD patients over the years. Based on Thomas Yu's brain pathylation, we found that there is community structure reconfiguration in both AD and BV-FTD at the whole brain level. When we narrow down to specific networks, the two disorders have differential network reorganizations. Specifically, Alzheimer's disease had more fragmental modular structure in the default and executive control network, while the BV-FTD patients had more severe damage of the modular structure to the salience and subcortical structure. Our findings support the distinct loss of network modularity in AD and BV-FTD. We have also shown previously that across the BV-FTD patients, the reduced salience network functional connectivity at the right frontal insula was associated with disease severity measured by the clinical dementia symptom. Now, by considering inter-network functional connectivity, we found that the increased salience network participant coefficient, indicating the loss of functional distinctiveness related to a more severe neuropsychiatric inventory score across all patients. When we focus on Alzheimer's disease alone, recently, several works, including Gil Rabinovic's group, have tried to explain the clinical variability in AD using functional connectivity mapping. However, this study only examined AD patients without comorbid conditions. As you know, AD frequently coexists with cerebral vascular disease. In fact, AD with cerebral vascular disease has become the most prevalent dementia subtype, especially in Asia. On its own, cerebral vascular disease is associated with decline in processing speed and executive function. Previous study in imaging as well as pathology have suggested that the cerebral vascular disease and AD could have additive effects on cognitive decline. For the same clinical dementia severity, AD-only patients present with more severe AD pathology than those comorbid with cerebral vascular disease. So, Joanna Chong, PhD student from my lab, asked this question: would we see a similar or different functional connectivity patterns in AD patients with and without cerebral vascular disease? Based on the memory clinical cohort from Singapore, we show that AD patients with and without cerebral vascular disease featured divergent functional connectivity changes in executive control and default mode network. AD patients showed reduced functional connectivity in the default mode network between PCC and posterior DMN. In contrast, the AD with CVD patients do not have any changes in the default mode network. In the executive control network, both AD with and without CVD had decreased frontal-parietal connectivity. Importantly, as highlighted in the yellow color here, only AD with CVD patients have increased frontal executive control network connectivity. And such pattern were mirrored very nicely in participants with mild cognitive impairment, no dementia. When we compare AD with and without CVD head-to-head, we found that AD patients have more reduction in the deformal network, which was related to the lower hippocampal volume. Similarly, when we look at the CVD groups, they have higher frontal executive control network connectivity, which was correlated with greater burden of white matter hyperintensity, a marker of CVD. Taken together, these findings lend further support to network-based dysfunction hypothesis at the dementia stage. However, we know that the pathological process is evident in the brain before cognitive or functional decline is observed. Given multiple failures of treatment development in dementia, it is crucial for us to target the preclinical or prodromal stage of the disease so that we can intervene early before any irreversible changes take place. So, can we detect brain functional connectivity changes and track them longitudinally in pre-dementia stage? Build upon our previous work described earlier, in collaboration with Juan Zhou from South Korea, we are interested in whether amyloid and cerebrovascular burden are associated with differential network connectivity changes over time in patients with mild cognitive impairment. So note that this is a longitudinal study, not cross-sectional. As expected, high amyloid burden, marked by the PIP-positive markers here, showed a reduction longitudinally in the deformal network connectivity, as highlighted in connection in blue color. Well, this is not true for the PIP-negative participants. In contrast, in the executive control network, on the left side, the amnestic MCI participants shows a mixture of increases and decreases in this network connectivity over time. However, the subcortical vascular MCI, regardless their amyloid status, they showed a functional connectivity increases in the executive control network over time, which is consistent with our brain study. These findings suggest the divergent influence of amyloid and cerebrovascular burden on the longitudinal trajectories of functional connectivity in these two networks in pre-dementia stage. Now, if we look at an even earlier stage, preclinical stage, most of the previous resting state fMRI study is based on cross-sectional design. How these network changes lead to cognitive decline over time and the influence of risk factors such as ApoE genotype in healthy elderly remains largely unknown. To address this question, my postdoc fellow, Dr. Eric Ng, studied the longitudinal functional connectivity changes over 4 years in a group of Singaporean healthy older adults aged 50 and to 85. We hypothesized that the integrity of the intra- and inter-network functional connectivity in 3 higher-level cognitive networks would deteriorate over time and relate to the longitudinal cognitive decline. Using linear mixed modeling, here are the results. Each line here represents one participant. And what we found is that aging is associated with decline in functional specialization in deformal network and executive control network. It's not the case for salience network. When we look at the inter-network functional connectivity, the y-axis here represents the connectivity between the executive control and deformal network. Higher connectivity here represents lower functional segregation between the two networks. As you can see, aging is associated with age-dependent decline in the functional segregation between the two networks. And more importantly, when we extract the rate of change from functional connectivity and processing speed, we found that greater loss of functional segregation between these two networks on the y-axis was associated with faster decline in processing speed. Processing speed is in the y-axis and the functional connectivity in the x-axis. So this is the relationship of their rate of change over time. Interestingly, when we divide the participants into ApoE4 carriers and non-carriers, we found that ApoE4 carriers in red had faster loss of functional segregation over time compared to non-carriers in green, suggesting that ApoE4 carrier had an accelerated aging pattern. Besides those two questions, under the framework of network-based dysfunction, we also hope to predict disease progression, monitor treatment outcome, and review disease mechanism. Our previous work has demonstrated this possibility at the group level. Specifically, we show that from healthy functional connectome, regions showing shorter functional paths to the disease epicenter and higher connectivity to other regions in the network had greater atrophy in disease. And this is true for all 5 disorders, which fit best with the transneural spread model and nodal stress model of the network breakdown mechanism in neurodegeneration. Although this study suggests that healthy brain functional connectome can give us some insight on the region-by-region vulnerability, this is obviously just the first step. Moving forward, we need longitudinal multimodal neuroimaging study, combining structural and functional, to more formally test the predictions made by various disease spread models and detect the early onset regions. We also need to combine imaging with blood, CSF or amyloid, PET imaging or tau, as well as behavioral or environmental factors to tackle cohort heterogeneity, detect early onset, and improve stratification. This will facilitate prognosis at the individual level. And of course, breakthroughs in machine learning, computational and statistical analysis are necessary to discover more robust and valid features for us to achieve higher accuracy and generalizability. And lastly, as disease-modifying treatments emerge, reliability and reproducibility of neuroimaging assay with feasible clinical parameters should be established. With that, I'd like to thank the funding agency and all the collaborators, students and postdocs who contributed to these studies. Here's the list of posters from my team, and you're welcome to visit us, and thank you for your attention.